excretion and elimination of waste products. Yes. First of all, what is the excretion process? The process of elimination of nitrogenous waste or metabolic waste is called excretion. Excretion is carried out by excretory system in human being. The major organ in excretory system is kidney. Okay. Yes. What are waste products? This is, imagine, cell nucleus. There is a continuous formation of, okay, ammonia, urea, uric acid. Okay, these are all are metabolic waste or excretory waste. These products should be completely eliminated or partly eliminated to excretory system. Here, these metabolic waste are formed from where? Usually, these metabolic wastes are formed by two ways. That is, one is excess ingestion. Okay, excess ingestion, and one more is through metabolism. Through metabolism, if we take excess food amount, that converts into metabolic waste or excretory waste. Clear? Understand? Yes. What are the major source for these? Formation of excretory waste means protein, ammonia, nucleic acids, nitrogen bases in that ammonia is present. Based on excretion of nitrogenous waste, animals are categorized into various types. That is, number one, ammonotelic, and the mechanism we are calling as ammonotelic. What is ammonotelic animals? Those animals excretes their nitrogenous waste, chief nitrogenous waste as ammonia we are calling as ammonotelic animals, and mechanism we are calling as ammonotelism. Examples for this. Say, what are the examples for this? Bony fishes. Means nothing but osteoptis. Okay, understand? And also many aquatic amphibians, aquatic arthropods, aquatic molluscans also. Reason for the elimination of ammonia, large amount of water is required. In fact, say the type of excretory products is mainly depend upon number one, habitats, number two, complexity of organism, and demand or perfect condition, particular condition. Now what I am telling, based on habitat, these are aquatic organisms require more amount of water for the elimination of ammonia. Now the question is, ammonia is formed from where? Ammonia is formed from liver through ornithine cycle. Ammonia is formed in liver, is readily to soluble in water or nothing but blood. Okay, understand? Clear? Yes. This is enter into the blood and excrete through urinary waste or excretory waste. Ammonia is eliminated as ammonium ion in fishes through gills. Remember, in the excretion of ammonia, the role of kidney is nothing. Very important bit. Okay, next. Ureotelic animals. Ureotelic animals. Ureotelism here. Okay, here based on habitat, ureotelic as well as uricotelic animals mostly terrestrial habitat animals. Understand? Yes, for this, best example, conrectis cartilaginous fishes. The process of elimination of urea as a chief nitrogenous waste. Animals we are calling as ureotelic animals, and the mechanism we are calling as ureotelism. Examples for this. Yes, conrectis. Conrectis. Okay. Terrestrial amphibians. Terrestrial amphibians. Okay. Understand? These are some examples. Even mammals, birds, sorry, not mammals only, including human beings, are examples for ureotelism or ureotelic animals. Urea is 1 lakh times lesser toxic than ammonia. Okay, in the elimination of urea, kidney plays a major role. Small amount of urea reabsorbed by renal tubules of the nephron to maintain the desired osmolarity in the medullary interstitium. Okay, next, uricotelic animals. Uricotelic and uricotelism. Those animals excrete their chief nitrogenous waste as uric acid. Uric acid, right? Understand? We are calling as uricotelic animals, and mechanism we are calling as uricotelic animals. Just example for this. Nana, land insects. Okay. Next, terrestrial habitat containing animals, and also that say birds, reptiles. Here, very small amount of water is required for the elimination of this uric acid. In fact, birds excrete their uric acid in the form of small pellets. This is a process to reduce the conserve the water, because these are all terrestrial habitat containing of farming animals. Understand? This is regarding categorization of animals based on their nitrogenous waste or excretory waste. Remember one more, aminotelic. This is very small one. We are observing in few animals only, not ammonotelic. It is an aminotelic. Aminotelism rarely we are observing in fishes only. Right? Nana, the type of excretory product eliminated by certain individual organism that depends upon phase or stage of organism. Okay, and type of habitat and condition. Best example, say arthropoda, otherwise annelida, earthworm. It is an aminotelic when the more water is present. Right? Next, this becomes into ureotelic when water availability is less. Next, toad, it is an amphibian. Larval stage, it is an ammonotelic. When it will become adult, this is an ureotelic. It's true that the type of excretory product depends upon habitat. Even toads, when the water is less, that converts into ureotelic to conserve the water. Okay, understand? Yes. Let us talk about now human excretory system. Human excretory system. Human excretory system consists of various parts, which includes either paired structures and unpaired structure. Let us talk about paired one as well as unpaired one. Paired one, a pair of kidneys, right? And ureters, a pair of urinary bladder and urethra. Let us talk about first physiology of kidney, external appearance of kidneys. Kidneys usually reddish brown in color. Reddish brown in color. Bean-shaped structure. The shape of the kidney is bean-shaped. Position of kidneys. Where we observe a pair of kidneys are present on either side of the vertebral column on the dorsal side of the body in between last thoracic vertebra and third lumbar vertebra. We consider this one, say T12 and L3. This is the, we may got the question like this: 212 last thoracic vertebra and third lumbar vertebra. Yes. What are the measurements of these kidneys? Kidneys usually say 10 to 12 centimeters in length. Next, 5 to 7 centimeters in width. Okay, 2 to 3 centimeters in thickness. What about the weight of kidney? 120 to 170 grams in adult healthy human being. Remember, kidney is very close to the, okay, dorsal side of the abdominal cavity. Okay, dorsal inner side of the abdominal wall as peritoneal organ. That's what kidney we consider as retroperitoneal organs, because one side only peritoneal or layer covers on the kidneys. Okay. Yes. In each kidney, 1 million, more than 1 million nephrons are present. Nephrons usually, okay, structural and functional unit of kidney, but these are only functional unit of kidneys. Functional unit of kidneys. Yes, fine. Let us draw that, say, LS of kidney, longitudinal section of kidney. First, let me complete that diagrammatic representation of excretory system. So this is left side, right side, and the right side of the kidney is always somewhat lower than left side of the kidney due to the presence of liver. Liver is the largest organ in human body. Yes. Okay, na? Clear? Yes, this is kidney. Human excretory system consists of a pair of kidneys, a pair of ureters, urinary bladder, and urethra. Okay, na? Understand? Yes. Let us draw that longitudinal section of kidney. I am trying to draw expansion of one kidney structure by showing all anatomical structures of kidneys. Yes, usually in each kidney, two zones are there. That is outer cortex. Okay. Next to middle concave. Okay. Understand? Say outer convex to middle concave. Middle concave. Now say inner surface of the kidneys, a deep notch is present. We are calling as hilum, through which through which say renal arteries, nerves, okay, renal arteries, that is blood vessels, arteries and veins and nerves and ureters emerging out, coming out. Okay, now clear? Yes. Now inside the hilum, a funnel shaped structure is present. We are calling as renal pelvis with a calyx calyces like this. The surfaces, that is outer peripheral region, that is cortex. So middle portion we are calling as medulla like this. Right? Now say cortex is extend in between. This is in the medullary position. Our medulla is extend as conical shaped structure. We are calling as renal pyramids. Renal pyramids. Okay, understand? The cortex is extend between the medullary pyramids as renal columns or columns of Bedini. Columns of Bedini. Entire kidney is covered by tough renal capsule. Renal capsule. Let us draw the structure of nephron. Let us draw the structure of nephron. More than 1 billion nephrons are present in the human kidneys, in each kidney. Yes, nephrons are functional unit of kidneys. Each nephron has two structures, that is glomerulus and renal tubules. Renal tubules. Yes. What is glomerulus? Glomerulus, fine efferent arteriole, renal artery. Okay, renal artery forms a network of structure we are calling as, okay, glomerulus. Glomerulus is helps in filtration of blood. Let us draw the diagrammatic representation here. Yes. Now this is, yes, this is efferent arteriole. The diameter of the efferent arteriole is always larger than diameter of the efferent arteriole. Efferent arteriole forms the glomerular structure, whereas this efferent arteriole forms the vasa recta. Vasa recta means how, say, find peritubular structures around the say U-shaped structure of English loop. We are calling as vasa recta. Here, this renal tubules are formed by cup-shaped Bowman's capsule. Okay, cup-shaped Bowman's capsule. Okay, glomerulus along with Bowman's capsule we are calling as Malpighian bodies or Malpighian corpuscles. Next one, this is considered as PCT. This is DCT. This is Henle's loop. Okay, this is descending Henle's loop. This is ascending Henle's loop. This is we are calling as CT, collecting tubules or collecting ducts. Yes, now Bowman's capsule extended as highly coiled structure we are calling as proximal convoluted tubules. Proximal convoluted tubules extend as air-pin structures with descending English loop and ascending English loop. The ascending English loop is continuous as distal convoluted tubules. Here less foldings are there. Okay, many number of nephrons are opens into okay collecting duct. Usually glomerulus, Bowman's capsule, PCT, DCT is present in cortex region, whereas Henle's lobe is present in medullary region or deeper medullary region. But this collecting duct or collecting tubules starts from cortex region and enters into deeper regions of medulla, inner medulla, right? Yes, this is regarding say structure of nephron. Nana, based on presence or absence of vasa recta or the length of English loop, nephrons are of two types. Number one is juxtamedullary nephrons. Number two is cortical nephrons. Those nephrons, the English loop is not deeply entered into the inner medulla, are only present in the cortex region or outer medullary region we are calling as cortical nephrons. Most of the nephrons in our body are juxtamedullary, cortical nephrons. What are juxtamedullary nephrons? Means the Henle's loop is deeply entered into the inner medulla. Is it right? So such a nephrons we are calling as juxtamedullary nephrons. Juxtamedullary nephrons. Here the vasa recta is highly reduced or absent in cortical nephrons. Cortical nephrons. This is regarding structure of nephron. Let us talk about urine formation. Urine formation. Let me draw the diagrammatic representation here. This is Bowman's capsule structure. Finish. Urine formation. Urine formation. Three major step process. Three major steps process. That is, number one, glomerular filtration. Number 2, tubular reabsorption. Number 3, selective secretion. Selective secretion. Let us talk about this one first. Glomerulus. Right? You know that glomerulus is formed by efferent arteriole. Efferent arteriole. This is an renal artery which supplies the oxygenated blood. Clear? To the kidneys or nephron here in this glomerular filtration. First, the entire composition of plasma as it is filtered into lumens of Bowman's capsule, so that this is called where filtration is going on, glomeruli. That's what we are calling as glomerular filtration. Glomerular filtration. And entire plasma is entering into the lumen of the Bowman's capsule except plasma proteins. That's what we are also calling this one ultrafiltration. Ultrafiltration. Now, how the glomerular filtration occurs? What is the reason behind that? Through which filtration membrane? How many layers are there? Let me explain. Actually, glomerular filtration occurs because of NFP, net filtration pressure 10 mmHg. When the blood is floating through efferent arteriole, glomerular hydrostatic capsular pressure, this is GHCP, is 62 mmHg, which is opposed by colloidal osmotic pressure and non-filtrate hydrostatic capsular pressure as well as colloidal osmotic pressure. Now, say glomerular hydrostatic capsular pressure. This is over. Next one, colloidal osmotic pressure, that is 32 mmHg. Next to capsular hydrostatic pressure, that is 18 mmHg. These two will oppose, so that 62 minus 32 plus 18, so that will get 10 mmHg because of net filtration pressure in the that is 10 mmHg in glomeruli. Right? The filtration of blood easily occurs through glomerulus. What about filtration membrane? Filtration membrane is formed by 3 layers. What are that? Say first one, this one, say endothelium of glomeruli, basement membrane, epithelium of Bowman's capsule. First one, endothelium of glomeruli, basement membrane. Next, epithelium of Bowman's capsule. Observe here, very important nitbits. 3 layers are present, but 2 only cellular layers. Basement membrane is a cellular layer, remember. This is applicable in diffusion membrane also. Observe here, epithelium Bowman's capsule has a specialized cells. They are called podocytes. Which are arranged in intricate manner, called slit pores. Say like this, through which the blood is easily filtered into the lumens of the Bowman's capsule. This one we are calling as primary urine. Primary urine. Primary urine is hypotonic to blood plasma. Now how much filtrate will form? Nana, almost 1100 to 1200 ml of blood is filtered by both kidneys per minute, which is equal to, okay, 1 by 5th position of, okay, 1 by 5th position of cardiac output, contractile ventricles of cardiac output. Also, this is an equal to 20 percentile of cardiac output. Reason? 5000 ml cardiac output per minute. What about this one? 1100, 1 by 5th position. How much filtrate is formed means we are calling as GFR. The total filtrate formed by both kidneys per minute is 125 ml per minute. Per minute, 7.5 liters per hour, 180 liters per day. Remember, the glomerular filtration rate is efficiently regulated by GGA, juxtaglomerular apparatus. When GFR is reduced, or blood pressure is reduced, or body fluid volume is reduced, immediately this activates the GGA, juxtaglomerular apparatus. Juxtaglomerular apparatus means, okay, say a special region present in between efferent arteriole and DCT where they contact together, juxtaglomerular apparatus. Whenever JGA is activated JGA releases the or stimulates to release the renin from efferent arteriole into the blood. Released renin acts on angiotensinogen. Angiotensin 1. Angiotensin 1 is converts into angiotensin 2 with the help of angiotensin converting enzyme. This angiotensin converting enzyme released from lungs. Now, angiotensin 2 is powerful vasoconstrictor, so that increase the glomerular blood pressure, so that increases the automatically glomerular filtration rate. Glomerular filtration rate. Now, what I told you here, say per day 180 liters of urine is excreting. Urine is excreting. Is it right? Then actual urine of excretion per day by adult healthy human being is 1000 ml to 1500 ml or 1 to 1.5 liters only. That's what here major amounts of, okay, almost 99% of the filtrate reabsorbed by the renal tubules at various segments, that may be PCT, okay, Henley slope, DCT and CD collecting ducts. Okay. Usually this first, say tubular reabsorption. Tubular reabsorption is selective process. Tubular secretion is selective process, but glomerular filtration is non-selective process. Glomerular filtration is a passive transport mechanism. This tubular secretion and selective secretion, it may be active transport mechanism or active process or passive process. Passive process, right? Understand what is the importance of tubular reabsorption and selective secretion means both together maintains the pH and the ionic balance of the body fluid volume. Is it right? This is regarding urine formation. Nana, here only I am telling urine formation is equal to glomerular filtration minus tubular reabsorption plus selective secretion. minus these two plus. Okay, tubular. Okay, urine formation is equal to glomerular filtration minus tubular reabsorption plus selective secretion. Selective secretion. Understand? Let us talk about the functions of the tubules. Functions of the tubules. Nana, first let us talk about PCT. This is PCT. Okay, flowing like this filtrate, curve towards right. Finally, here let us talk about here. PCT. PCT is lined by simple cuboidal epithelium, breast border epithelium, which increase the surface area for reabsorption. It is a major site for reabsorption. In this PCT, nearly all essential nutrients, okay, and 70% of electrolytes, that is NaCl, H plus ions, okay, K plus ions, potassium ions, okay, are reabsorbed from this PCT only. PCT helps in maintenance of pH of blood, that is 7.2 to 7.4, by selective secretion of H plus ions, ammonia ions, okay, and K plus ions into filter bicarbonate ions. Sir, what is the link to the segregation of H plus ions and reabsorption of bicarbonate ions? This is might be recalled from breathing and exchange of gases. We studied well at the time of what do you call transportation of carbon dioxide. Okay, chloride shift. ಬೈಕಾರ್ಬೊನೇಟ್ಸ್ ಆಯನ್ ಶಿಫ್ಟ್ ಆಗಿದಂತೆ NaCl HCl ಶಿಫ್ಟ್ ಆಗ್ತದೆ. ಇದು H ಪ್ಲಸ್ ಅಯಾನ್ ಜಾಸ್ತಿ ಆಗಿದಂತೆ pH ಕಡಿಮೆ ಆಗ್ತದೆ. pH ಕಡಿಮೆ ಆಗಿದಂತೆ H plus ions ಜಾಸ್ತಿ ಇರ್ತದೆ. This is a link. Next, let us talk about Henle's loop up to here. This one. Nana, say Henle's loop. Two portions are there. That is descending Henle's loop, ascending Henle's loop. Fine. Clear? Let us talk about this one. First, descending Henle's loop. Hairpin structure. Okay. Henle's loop function is increase the or maintains the high osmolarity towards the say medullary interstitium. This is inner medulla. Yes, okay na? Understand? Clear? Yes. Descending Henle's loop is permeable to water through osmosis process, that is passive transport mechanism. It is highly impermeable to electrolytes, so that as the filtrate moves down towards the inner medulla, the concentration of the filtrate is increased. Now let us talk about this one. Descending Henle's loop. The reabsorption in ascending portion of Henle's loop is minimum. This ascending Henle's loop is opposite to descending Henle's loop. This is permeable to electrolytes, okay, and impermeable to water. Nana, these electrolytes may reabsorb up, okay, right, by either active or passive transport. Electrolyte, we take one example, NaCl from the thin segment of ascending portion of Enlist loop by passive transport mechanism, whereas from thick segment of ascending portion of NaCl by active transport mechanism. Okay, next DCT. In DCT, conditional reabsorption of sodium and water takes place. Sodium and water takes place. Nana, DCT plays a major role in pH and sodium, okay, balance, sodium ionic balance of body fluids by selective secretion of H plus ions, ammonia ions, okay, and K plus ions into filtrate, and reabsorption of bicarbonates ions from filtrate. Conditional reabsorption, usually the reabsorption from DCT is mainly depends upon antidiuretic hormone. Antidiuretic hormone secreted from neurohypophysis of, okay, pituitary gland, also called as vasopressin, which is a powerful vasoconstrictor. Okay, yes, which acts on later part of nephron, nothing but DCT, okay, and helps in conservation of water, nothing but indirectly prevents the loss of water. Antidiuretic hormone. Diuresis means water loss. Okay, na? Understand? This is regarding DCT. Next, let us talk about collecting tubules or collecting ducts. Collecting duct is extended from cortex to inner medulla of kidneys. Collecting ducts helps in maintenance of pH and ionic balance of body fluids volume. Both do same functions. By selective secretion of H plus ions and K plus ions into filtrate, and also small amount of urea is also reabsorbed into medullary interstitium from collecting duct to keep up high osmolarity in medullary interstitium. Okay, this is regarding functions of the tubules. Let us talk about the mechanism of counter current or counter current mechanism. Okay, na? Can we continue?